So now I think we can start with our first presenter, Georg Tremmel. Georg Tremmel is an Austrian artist who currently lives in Japan. Being a member of Biomedia Group in Waseda University in Japan, Georg Tremmel is a part of a framework, BCL, which conducts artistic research. And the title of Georg Tremmel paper today is Ghost in the Cell, Synthetic Heartbeats. Please welcome with me Georg Tremmel.
Thank you, Olga, for the kind invitation, the kind reception. So you basically already, my slides are visible, right? Yeah, that's always the thing. So you basically already talked about myself, but just to help people further contextualize myself. So I have an art background. I studied media art in Vienna. I also studied at the Royal College of Art in London. With Shio Fukuhara, I started at BCL, which you said is an artistic research framework, investigating the borders between art and biotechnology. And I also was working for 10 years at the University of Tokyo in a laboratory for DNA information analysis, actually not as an artist, but as a special project researcher, just because I wanted to experience the culture of natural science and wanted to immerse myself. But I'm very happy I got out there now. So I'm actually also now a PhD student at the University of Applied Art in Vienna. So it's a remote situation, but I think we're all used to this now. And you kindly mentioned metaphors already. And if you ever come to Tokyo, please visit us also at the BioClub Tokyo, which is our local biohacker space, which I founded like about 2 years ago. Some of my projects, which I will just mention, just to give you a very quick idea. So our first project was called Biopresence, which was still done at the RCA, where we first proposed and then we encoded human DNA within the DNA of trees, but we developed a special method without changing the phenotype of the tree, but still allowing the human the information of the human DNA to live on within the DNA of a tree. So this was done nearly 20 years ago now. Another project we are maybe well known for is called Common Flower Commons, which deals with genetically modified blue carnation and their status, their legal surroundings, and whether it's possible to create a private copy of these GMO flowers. A development of this work is called Whiteout, where I think it was one of the first artworks which used CRISPR about 7 or 8 years ago, where we used CRISPR to remove the blue gene responsible for the blue color and turned the blue flower white again. Okay, there's a typo, never mind. So resistive use deals with the history of biological warfare, especially from the position of Japan. And the Blacklist printer is a work situated on the border between synthetic and analytical biology. It's a proposed home DIY printer which only prints forbidden DNA sequences like viruses and so on. This was done like 2 or 3 years ago, and now, of course, it's becoming a bit difficult to experience to exhibit this. So an upcoming project is Copyright Healer, where we question the agency and the legal status of tissue cultures, in this especially case the living tissues of Gila Henrietta Lacks, and question whether it's possible for her to take a picture and obtain the copyright of this picture. But this is ongoing. But now to this talk. Again, some background slides. So we all know the 20th century was the century of the computer, and the 21st century is already the century of biology. When I started showing these slides like 15 years ago, I always had to say it will be the century of biology, but now we know it already is. So with the revolution that CRISPR started, with kind of the whole genome editing, with the renaming of the Human Genome Project to Human Genome Project Read, and the advent of the Human Genome Project Write. So we are on the verge of very big changes for not only for our species but for our whole planet. So I'm very interested in this border between the analytic and the synthetic. And as we know, biology was until recently a purely analytical science. So we could only observe, we could only classify, we could create systems. But since the advent of targeted genome editing, this changed from read to read-write. And of course, we all know the word synthetic biology. But now with the precision synthetic biology, it's possible to change, everything we can think of, or is it? So I also kind of always would like to show this slide. So this is a book by this is a slide from a book by Donatan and Fiona Raby, who I had the privilege to be students with at the Royal College of Art. And in their speculative futures, they had this taxonomy of futures. So what kind of futures are there? There are preferable, probable, plausible, and possible futures. And they argued it's our duty as artists and designers to not only imagine these futures, but to create discussions about these different kinds of futures with the aim to kind of prepare society for these futures. But funnily enough, they did not come up with this time beam, but this was done in 1994 in a healthcare journal, which actually the quality is a bit bad, but you hopefully can see it's actually a three-dimensional time beam. But what Tony and Fiona did not include was this wild card. Yeah, so the unknowns. So what are the things we don't know yet? And of course, that's the problem of predicting the future. You're not really sure what it will be. Yeah. So, but our projects always they always happen in these shared spaces and the shared codes between biological and digital life forms, or between the carbon and the silicon. And why are we called? So when I speak of we, I always say BCL. So BCL is an homage to the BCL research group, which was started and run by Heinz von Förster. Heinz von Förster is an Austrian-born cyberneticist. So he introduced the cybernetics of cybernetics, and his research group was called the Biological Computer Laboratory. And this started in 1959 and was active until 1974. And he fostered a culture where not only scientists, but also philosophers and artists could collaborate and work on common problems. And I still believe that, Heinz von Förster still has a kind of unknown, but a big role to play in the advent of early artificial intelligence. And I think hopefully his role will be more honored through time. So I can recommend these books. He actually edited the Macy Conference. It's the famous series. So we are also not a group, but we are more an artistic research framework. I have to admit, I used the word artistic research before I actually knew what artistic research meant. But becoming now an artistic research PhD student, I'm more aware of what it is. But still, maybe that's a good thing about this term. There are many different definitions of what artistic research can be, so we don't have to dwell only on one. So about this work. So Ghost in the Cell, Synthetic Heartbeats, this is actually a development of a work we did about already 5 years ago. And I will tell you the background, and then it's easier for you to understand the current work. So I'm curious, is Hatsune Miku, is she famous also around the world? So I know she's very famous in Japan. I'm not quite sure how famous she is in other Asian countries. But so Hatsune Miku is a so-called virtual idol, which started out as a synthetic voice. So it was a so-called Vocaloid, a software package. So on the right, you can see like the synthesizer on the left. This is the package of the synthesizer, which features this drawing of this manga-like girl with this very green hair. So, but what happened? The company who produced this software encouraged the community to remix to make remixes not only of her voice, but also of the images of the character itself. And through this act of releasing the usage rights of this image to the wider community, she gained not only a following, but she also nearly gained a life on her own. So her voice was many times in the charts. So she created not only music videos but only live performances, and so she became a bona fide pop idol here in Japan. But it's not. The interesting thing is it's a community effort. So it was edited by the community. Of how much it was instrumented by the company is a different question. But I don't think it was their purpose to create this. It really emerged poetically. So when we came across this, so the interesting thing was, of course, she has a voice, she has an image. I don't know if she has a soul, but she surely doesn't have a heart, because she's a constructed culture. But I'm sorry. But what we did, we thought if it might be possible to create a heart for her, and how could we do that? So we had some experience with in vitro culture of mammalian cells, and it was possible. Sorry. I'm sorry. So it was possible to use Japanese tissue samples from a female donor, and we asked the community to create a synthetic genome. So we had a little website where it was possible to change the DNA code purely on a computation, on a text basis, and this DNA as a text we then included into the iPS cells. Which included the iPS cells, and these iPS cells were then differentiated into so-called cardiomyocytes or heart cells. So what we did, we created cardiomyocytes with the DNA with the community source DNA of Hatsune Miku. So it was quite an interesting challenge to exhibit this work, and we were happy to have the 21st Century Museum as a collaboration partner. And so this was the design of the work. This was more design, and this is how it looked in the installation piece. So it was basically a big empty room with an incubator in the center and a screen to enhance the tissue cultures in the. So just to give you an idea of how it did look like. So it was, of course, it was some sort of experience, and we actually used, this was 2015. So this was the hype of Deep Dream. So we used deep dreamed images around this, around the exhibition space to symbolize the dreams that Hatsune Miku's tissue might have, going between living and going between states of living and death, or being in this state of semi-living. So what is interesting about cardiomyocytes is that they unlike other cells, they start to beat automatically on their own. So whenever they create a certain, whenever they're grown to a certain stage, they start beating on their own. I'm sorry, I cannot control this sound now, but I hope you can see this beating. So this is a live installation view of the heart cells. So this is one heartbeat. And we came into the position of having or being approached by collectors to actually collect this artwork, and we had a long discussion of how we can actually how we can actually create a collectible artwork from this piece, because that's a problem with art and biology or bioart. It's really difficult to not only to collect but also to archive. And of course, you can say on the one hand it can be a performance, and this performance can be archived. But for this, we decided to release single heartbeats. So the collector could collect one single heartbeat, which is looped here, and this is about 70 frames, and this is one heartbeat, and this is how this work manifests itself in collections and in museums nowadays. And now, finally, to the sorry for taking up so much time. So finally, for this project now, so synthetic heartbeats. I was very, like many artists in the last couple of years, was very much inspired by the image-to-image translation networks, and especially by the Pix2Pix paper by Isola et al. And of course, we know how this works. And so this is of course another famous example. And I was challenging myself if it's possible to actually generate synthetic heartbeats of this work, which would be indistinguishable from the real heartbeats. But of course, I was a bit of the disadvantages, because although I have some footage of the video footage, but I don't have access anymore to the actual. So restaging the work would require a lot of resources again. And actually, that's a funny detail I did not mention. So at the exhibition. Because it was a bit further away, so we couldn't go there every day. So the curator, so Takashi-san, so he had to learn how to keep the cells alive, yeah. So how to keep these iPS cells alive in an exhibition space, and he had to feed them every day, and he became very good at it. So this was quite an interesting side thing that I forgot to mention before. So I was challenged myself to do this, and I do have some preliminary results, but they're not really where I want them to be. So I would just present them for discussion at this stage. And of course, the question is also, as we know, it's really hard to create high resolution images and not only low resolution. So there's still a lot of work to do in this stage. But I will be curious. So what I would really like to do is how to integrate this actual synthetic DNA into a machine learning model, and kind of really entwine these spaces of the biological and the digital. And of course, the question is then, how will the digital DNA codes manifest itself in the biological models, and or will they, or how should they manifest itself? Okay, so I think I'll leave it like this. So I just have to thank my collaborators, Shia Fukuhara, Philip Boeing, Yuki Yoshioka, Takashi-san from 20th Century, so now Doku, who spent a lot of time with me in the machine learning design and semi-transparent design, and of course the 21st Century Museum in Kanazawa. And if you have any further questions, please do get in touch with me. I'm happy to exchange. I hope I was too fast, maybe. Yeah.
Thank you, Georg. You were right on time.
Okay, great.
Thank you for your presentation. And now we have 1 minute for our online attendees to type their questions via Zoom chat.
Okay.
The chat room will be closed after 1 minute, and the next presentation will begin then.
Okay, so we'll do general discussion afterwards, if I understood this correctly.
Yes.
Yeah. Okay. So shall I exit?
Probably you close. Yeah.
Okay.
And now we're moving on to our next speakers. Our next speakers are Maggie Roberts and Stephanie Moran. Maggie Roberts is an internationally established artist and co-founder of Orphan Drift that explores the boundaries of human and machine vision in an art practice. Maggie Roberts currently teaches critical studies at University of Arts in London. And Stephanie Moran is an associate partner at Ethic Lab, which is a design and technology research consultancy. Stephanie Moran is currently a PhD candidate at Plymouth University. The title of the speech of Stephanie Moran and Maggie Roberts is ISCRI, Communicating Between Two Alien Intelligences Through Art. Let's welcome Maggie Roberts and Stephanie Moran presentation.
Sorry, I was muted. 1 second. Hi, everyone. Thanks for the introduction. Can you hear me?
Yes.
Thanks. So, ISCRI is an experiment in communication between 3 species, cephalopod, AI, and human, and is part of a global conversation around shifting the agendas that currently determine AI evolution. Our collaboration itself is also an experiment, an interdisciplinary inquiry between multimedia and computational artists, machine learning technologists, cultural theorists, an interspecies communicator, a sociologist, and marine scientists. In development for over 2 years, ISCRI proposes training an AI that gathers data from sensors placed in an ocean mesocosm housing a common octopus. The AI system will evolve through the latest unsupervised learning models, ensuring that it has not learnt primarily from a human agenda, but from a fellow distributed consciousness, an octopus, operating in an uncertain and fluid environment, water. We're designing a space wherein the AI may communicate with the octopus in ways we cannot recognize. So maybe we don't need to introduce ourselves, Stephanie. So,
Yeah, I don't think we do. So ISCRI sees itself as part of a wider cross-disciplinary effort to destabilize the hierarching Western mentality that solipsistically places human values and survival above all other species. It sees aesthetics as integral to this. We define aesthetics as the capacity for taking pleasure or displeasure in an object or event, or engaging in activities that produce or seek sensory stimulation. Aesthetic behavior can be seen as a mode of cognition, a way of engaging with the world that extends beyond practical motivations such as the search for food or a mate. Non-human animals might therefore display aesthetic behaviors or engage in aesthetic relationships, and octopuses seem good models for non-human aesthetic behavior. They often appear to engage with the world in ways that extend beyond what is required for their individual survival or species reproduction. In animal biology, Burkhart's criteria for play align with our definition of aesthetics, that it is incompletely functional in the context in which it is expressed, that it is spontaneous and pleasurable, and that it differs from regular behavior. Marine biologists connect object play to exploratory behavior from which animals, including humans, learn. Kuba et al. Speculate from their research that play could be a byproduct of a complex nervous system heavily depending on learning, and have the function of maintaining a status quo in times of lesser ecological pressure and lack of stimuli. This may be evidenced in periods of intense aesthetic activity across human history. As octopuses are solitary creatures, there is little opportunity for social play, but studies show that they engage in playful interactions with objects. As in the video this image is from, the octopus moves around the object, touching it, prodding it, and trying to move it.
we've been observing octopuses in the Cape kelp forests, where the environment is home to myriad life forms, and all, whatever their scale and behaviours, move in response to the ebb, flow, and surge of the ocean. Nothing feels static. Even rock surfaces are covered in sponges, anemones, seaweeds. And the shallow depth of these coastal waters means that in daylight hours, the terrain is patterned by a complex play of light beams distorted and refracted by wave motion on the surface. The art team are translating and refiguring these phenomena into animated patterns and shapes. Recent research into cephalopod vision suggests that octopus and squid's U and W-shaped pupils enable vision based on chromatic aberration and light polarization by bending light. It's simplified. In addition, light receptors in the skin register specific waves that each color expresses, either absorbing or reflecting them. There are also mood triggers that produce a range of oranges, browns, purples, and reds in the skin as the basic structure of a language of feeling. We're generating objects, skins, and terrains in patterns of polarized light that simulate this intense spectral information and chromatic signature. We're developing what we're calling non-human aesthetics. Imagining a world where representation, perspective, central focus, horizon, and gravity are replaced by pattern, abstract flows of color and shape, haptic and tactile sensing, multiple simultaneous viewpoints and rhythmic pulses, visualizing the perception of a distributed intelligence that does not prioritize sight-led perception in the way a human does, but interprets the world as much through 360-degree touch, shifting light and dark, water pressure, and chemotactile information. Seen here, BBC underwater cameraman James Loudon is filming in extreme macro focus into crevices and cracks with a probe lens, capturing something of the tactile haptic world of the octopus. And Interspecies communicator Anna Breitenbach has helped us to develop a telepathic approach. Some of the computational art involves simulating octopus skin patterns using both Blender and Alan Turing's reaction diffusion coding. These are colored according to specific octopus moods: white for calm, orange, curious, blue, violet, red, excited or anxious. Chroma chatter, so-called, is probably display, perhaps involuntary streams of consciousness or dreaming, suggested in the 2018 documentary, The Octopus in My House. Other works abstract textural and shape detail in the environment into layered assemblages or generates geometric and synthetic, sometimes interactive, shapes and forms, developed in Unity and Houdini by George Sims. We may have an idea of the sort of effect coding will have, but the result is always a collaboration with an algorithm and involves recognizing and amplifying an outcome that unexpectedly manifests an idea. Art practice makes space for the unknown here, as procedural coding generates visions of possible embodiment, perception, and materiality, and increasingly of porous relations between the synthetic and organic. These are procedural and Voronoi textured octopus skins made in Blender by one of the computational artists, Megan Bagshaw, and I hope I'm getting all this right. She explains that essentially we're acting similarly to an AI, gathering a lot of data on, for example, what an octopus is visually or how its camouflage works. And once we can deconstruct the layers of event into mathematical and visual concepts, we can then feed those into something called a flow or node network in 3D animation. And Duncan, another of our computational artists, adds, we're noticing what's in the life world of an octopus: sine wave motion, fluid space, flocking behaviors, becoming the surrounding environment. And then we're using code to develop versions of nine-brained consciousness or flocking fluid environments that are generative and emergent. VFX and 3D softwares facilitate a radical expansion of the imagination towards the synthetic. Deborah Levitt writes in The Animatic Apparatus, Animation, Vitality and the Futures of the Image. Digital animation is a tool for moving away from the recognizable towards the unknown. It is expansive and questions subjectivity, gender, reality, materiality. It is viscerally intimate and neurospeculative. What we don't know can become a generative space. In Blender, physics are applied to objects to simulate gravity. The key moment for me was applying cloth physics to the wrong thing, a rock. This released me into the possible, to create layers of environment that float off each other, pixelate suddenly, or become protean, twisting and buckling as a seabed becomes a skin, perhaps more akin to octopus perception.
Cognitive scientist Frank Grasso defines cognition as any adaptive process which connects sensing in the world to action in the world, using adaptive in its cybernetic sense of a process which changes with experience. Current octopus cognition research is fundamentally changing our understanding of how octopuses perceive the world, from a better understanding of their chemotactile sense, that's a smell, taste, touch sense without a direct human equivalent, to the ways they may sense light through their skin and see the plane of polarization of light. In biological brains, networks of neurons process this information from the environment. Computational neural networks used in machine learning for AI are modeled on biological neurons, which are seen as the brain's processing power, although there are other elements to biological cognitive processes, such as biological pharmacology or its internal chemistry, neuroplasticity, electrophysiology, and synchronicity. Octopuses have around 500 million neurons, but more than half of those are in their arms. What does it mean for your intelligence to be distributed through your arms, to think with your arms? Octopus's suckers possess complex and sensitive sensors that convey environmental information, taste, smell, touch, feel, and movement, forming a spatial array that could be directed towards multiple objects in any direction, or clustered around and focused on one main object. Although, even when focused on one object, they tend to keep a couple of arms on the lookout for other sensory input. Surface contact is required for chemical and mechanical information to be transmitted, so each sucker samples a distinct location, producing a kind of sensory point cloud of information in aggregate. The neural networks in octopus arms are thought to coordinate sucker attachment and orientation with arm movement, as well as inter-sucker coordination. They seem to have the functionality of a second proto-brain with a high degree of autonomy. In our experiment, the octopus will be inhabiting a mesocosm or ocean-like environment, a little like this image. It will be equipped with underwater screens to intermittently stream the artwork and a wide array of sensors to detect changes in the environment. Sensors measuring, for example, color change, light, movement, and water pressure. Encouraged by famed octopus curiosity, we hope that it responds to the artworks. Any responses will be captured and passed by the AI artifact, which responds to and learns from changes in the rates of change in the environment that are detected by the sensors. Project ISCRI is premised on the understanding that we cannot know what it is like to possess such distributed cognition, but that another distributed cognition, an AI, may be able to learn from and communicate with the octopus if we do not preempt what that may entail. The concept of embodied cognition is important here. An understanding of knowledge and understanding as embedded in our environment and the ways we've evolved to perceive the world. We exist, all species exist, in ways that are deeply entwined with the environments we've evolved in. In reinforcement learning, the form of machine learning we've chosen for this project, the algorithm learns from unknowns. It's hard-coded to enjoy inhabiting a particular state and will try to return to that state as often as it can by manipulating its environment. Reinforcement learning motivates it to manipulate its environment in order to induce changes in itself. So in this case, the octopus is given the tools to code the AI itself as the artifact becomes a digital octopus in a digital environment. How might an octopus AI reflect the different cognitive structures of another very alien species?
So cut to two arms unfurl in the water, coiling gently in the drag and flow, inside and outside of the same temperature, dissolving your boundaried self. Colours flash in front of you, below and around you, sensing the chemical impressions of other creatures and multiplicities of intricate pressure. Sucker touch. Bursts of vision ripple outwards, then all of you coalesces in tight precision, registering a tiny prick somewhere in the expanded awareness. Detail anchors perception in the vastness. This world is endlessly relational. Your body the sometimes individual medium for processing it. Figure-ground relations fold into a mimetic, porous skin. Communication flows through current, colour and touch between enmeshed intelligences, communities and differences. At this point, we have a lot of questions in terms of the future use of the evolved artifact, and very interested in how it might change something of an experience in VR, but we'll need to work with VR development experts to answer many of our questions. For now, we can ask if VR can provide a platform for the octopus AI to in some way reproduce octopus-like behaviour that might help us experience to some extent and in intimate ways a consciousness alien to ours. Will the octopus AI respond to participants as if they were an octopus? And what might this entail for the kinds of embodiment possible in VR? And then for the possibilities of expanding what it means to be human through a VR-delivered multi-sensory synesthetic experience of non-human life world. Might we consider some form of communication with the AI thinkable in VR through the vehicle of its octopoid behaviour? Could an octopus AI modulate its VR environment to enable it to learn, like the octopus, to become tensile, reactive, and plastic in uncertain, fluid, and constantly changing environments that reflect real-world contexts?
So where are we up to now? We've spent 2 years researching octopus cognition and appropriate machine learning technologies. We're currently spending this year prototyping video materials, objects, sensor arrays in environments, and reinforcement learning algorithms. We've gathered a team of creative coding experts and a BBC underwater cameraman, James Loudon, an animal communicator and cult experimental electronic musician Steve Goodman, aka Code 9, and his record label Hyperdub. We've consulted with mesospasm specialists and cephalopod husbandry specialists, Graziano Fiorito at Stazione Zoologica Naples and Alex Harvey at the MBA in the UK. We're currently in conversation with a number of major international galleries regarding a touring exhibition in 2024. But and we're currently fundraising for the next phase, which is the Octopus and AI development work. This will be accompanied by a live stream and program of research and events. So we're working right now with the Serpentine Creative AI Lab and looking at a mix of foundation and corporate sponsorship, tech sponsorship, science, tech, STEAM research funding, art science awards, and gaming and tech investment, so that we can undertake the major next part of the project. Do get in touch if you'd like to know more, would like to host a leg of the touring exhibition, or can support the project in any way.
Thank you.
Thank you so much for a great presentation. Really a lot of input. So now we have 1 minute for our online attendees to type their questions via Zoom chat. The chat room will be closed after 1 minute, and we will move on to the next presentation. And while the questions are being typed, I'm going to talk about the next speaker to introduce her. So now our next speaker is Maro Pebo. Maro Pebo holds a PhD in creative media, and currently she is a senior lecturer in moist media in the Detao Masters Academy at the Shanghai Institute of Visual Art. Maro Pebo's talk is titled Microbial Emancipation. Let's welcome Maro Pebo.
Thank you, Olga, for the introduction. I have prepared a very short video, which is only 10 minutes, in which I will introduce the way that I'm working with not only the notion of interspecies, but rather the notion of transspecies as not something innovative or new, but something deeper in what we are biologically. So maybe you could play the video I prepared for you guys.
Hi, my name is Maro Pebo, and I'm an artist that works weaving collaborations to defy anthropocentrism and skeptical environmental accountability. My transdisciplinary work subverts the monopoly of the life sciences to think biological matter and aims to release the indiscipline of living cells. I have a PhD in creative media, and I specialize in the intersections of art, science, and biotechnology. My current interest lies in microorganisms cultures and in biological posthuman turn. Today, I will introduce 2 ideas. The first is the posthuman realization of symbiogenesis, the microbial origin of the mitochondria. And second, the use of VR, 3D modeling, and animation, as well as biotechnologies, to represent what can't be seen without mediation, to represent our relationship with microorganisms by imagining a language outside traditional life sciences representations. The work Microbial Emancipation was developed with Dr. Yun Lam and Malitzin Cortez, and it consists of a reliquary that holds the mitochondria from the artist's blood that used to be a free bacterium. Behind it, we will have a triptych. I'm showing this example. So we are talking about the same triptych format. This triptych displays a 3D model video where the history of symbiogenesis is narrated. So the side panels present the world as it has been for the majority of the history of life, a planet of microorganisms. The central panel of the triptych starts also with the microbial planet, but then continues to show how microorganisms ate each other until the occurrence of the event when an independent living bacteria engulfed by a bigger archaean did not get digested. It kept its genes and its independence to become the energy source of eukaryotic cells and be protected by them, now known as the mitochondria. In each of our cells, we have these ancient bacteria that are the source of our energy and still keep their own independent genome. This process was called symbiogenesis and was described and studied by Lynn Margolis. This is a remarkable fact that transforms the way we understand our human relationship with microorganisms. So we have known about the bacterial past of chloroplasts and mitochondria for decades, but this artwork wants to make it material, wants to have it happen, help us experience it in each of our cells. Microbial emancipation acknowledges and detaches this key organelle to remind us of the deepest relationship to microorganisms. how to materialize and show this ancient and invisible phenomenon? Well, for microbial emancipation, the premise is the liberation of the mitochondria in a desire to allow the now organelle to exist freely as a bacterium. This is no longer possible because of the number of genes it has lost in the uncountable generations of inhabiting other cells. But then, the idea of freeing the mitochondria becomes a ritual of proof, of undoing to demonstrate. For this purpose, in the lab of Juan Pedro Luna Arias at Simvestav, a research center in Mexico City, Gerson Vasquez helped me isolate the mitochondria from my white blood cells. So this is the first sacrifice, to give up my blood. In order to increase the number of white blood cells without needing a high amount of blood, on the advice of Dr. Jun Lam, we activated blastogenesis process in the blood samples, stimulating the reproduction of white blood cells or lymphocytes. When this process was finished, we continued with the mitochondrial isolation. The mitochondria are kept in an Eppendorf tube. A very small amount of transparent liquid is visible in the bottom, and this is tiny amount of matter that the mitochondria are. They exist again as an independent entity released from the containing engulfing cell. And then the notions of fate intersect religion and scientific experience. The Eppendorf tube was placed inside a model mitochondria hosted in a dedicated reliquary, designed in relationship to the tradition of Western reliquaries containing the remains of saints. In many cases, the shape of the medieval reliquaries echoed the part of the body or object contained, for example, an arm with a hand to hold a bone or a hand. In the case of microbial emancipation, the reliquary structure refers to the structure of the endoplasmic reticulum, which is in context and interaction with the mitochondria in the cell. And here we have the 3D modeled version. And this endoplasmic reticulum inspired reliquary was then 3D modeled, 3D printed to be casted in bronze. Here we have the 3D casted bronze version. And this is the rendering in the Herdau Museum in Brazil. So besides this physical version, Malitzin Cortez designed a virtual space to watch this video, where we opted for a spherical version to take advantage of the possibilities of VR. If someone wants to watch the video, we wouldn't want them to watch it as a flat surface, but as a virtual space, and then in virtual space, they could walk into sphere where the video and the story would be narrated. So this is the VR space that was presented at the Lahore Media Arts Festival in Pakistan. Concerning this video, we wanted to displace the theme of symbiogenesis from the realm of the life sciences. We were looking for a way to narrate the story of the mitochondria as a founding myth. We have developed a pop aesthetic that relates the shapes, the colors, the transparencies in order to present an alternative to common scientific renderings. We wanted cells, organelles, and bacteria to perform in a visual language that could transform our imaginaries of them. This is the language that we chose to make visible the invisible, to present the origin of our cells. Microbial emancipation participates in the transformation of the way we understand microorganisms. So there is a strong tendency to associate microorganisms to dirtiness, to disease, to think of them as undesirable entities only. And this association is challenged by the knowledge of the human microbiome and the role of microorganisms in all ecosystems. But this work pushes understanding of the relationship further, not only to display the enemy narrative or the narrative of microorganisms as guests working as our allies, but rather to trigger a reality displacement when we acknowledge the microbial origin of our cells and how still our organelles are so close to the bacteria that they used to be. The work then opens a set of questions, not only about our relationship with the microbial, but about our identity in general, about the way we understand individuals and the way that we divide species. Moreover, it gives recognition to the mitochondria. It gives recognition to the value of the mitochondria and to the sacred microbial origin of our metabolism. Thank you very much. Do you have any questions?
Thank you so much, Maro, for the video. As I understand, are you going to talk a little bit more, or it's the end, yeah?
Yeah, this is the end. I just wanted to invite people, if they have a chance and if they're in Hong Kong, to visit the gallery of the Art Machines exhibition so they can watch the whole video. But yeah, that's it. Thank you, Olga.
Yeah, thank you again. And now we have 1 minute for our online attendees to type their questions via Zoom chat. The chat room will be closed after 1 minute and the next presentation will begin. And now I would like to present our last speaker, last but not the least. Our next speaker is Park Tae-yoon. She is an artist and researcher. She is currently conducting her research with orchids at City University of Hong Kong. And the presentation of Park Tae-yoon is titled Environmental Machine Learning in Multispecies Agency, a case study of random forests. Please help me welcome Park Tae-yoon.
Thank you for your nice introduction. So yes, I'm going to talk about environmental machine learning in multispecies agency, a case study of random forest. But does everyone hear me very well? Okay. So I have been working on my art practice-based research about multispecies relationships and artistic engagement with eco-cinema. In this presentation, I would like to talk about my research and a case study of a random forest regarding machine learning and multispecies relationship. And secondly, environmental literacy in humans and non-humans. And thirdly, ecological correctness and ethical participation of art and technology. So since 2019, I have been researching plant-based multispecies relationships through my cinematic practice. I focus on multispecies relationships that are shaped by plants. I follow a few Hong Kong local orchid species and observe their life cycle to understand multispecies relationships and to reflect human species from plant-based perspective. So my choice of research method is artistic practice, especially with eco-cinematic practices. Eco-cinema also emphasizes the significance of multispecies relationships and ecological equity in cinema. Firstly, I started with filming and observing plants and multispecies interactions, as you can see in the slide. So those two species, left on the ant and also the right side in the ant with the wings. So they try to get in the orchid flowers. Somehow these insects were allured by the orchid flowers. So they get in to the flower and then the flowers would be pollinated with the movement and of the insects. So the main research objectives are challenging the human-centric gaze of the camera lens and filmmaking, and also visualizing plants' way of seeing the world. In exploring plant-based perspectives, I seek alternative methods to connect humans with multi-species dynamics to overcome the anthropocentric perspective in artistic practice. So ecocinema encompasses what it means to inhabit this planet, to be a member of planetary ecosystem or ecosphere, and to understand the value of this community in a systemic and non-hierarchical way. So I realized that the conventional way of filmmaking enhances human-centric gaze and started to doubt about so-called machine eyes. I see through camera lens when I film, and I began to raise questions such as, what is the role of camera lens? What's the position of the machines in the non-hierarchy of species and ecocinema? And does the machine mediate multi-species relationships? So to answer the questions, I have tried making a series of moving images. So on the left side, these examples show the progress of orchid seed capsule grows and gets mature. These moving image pieces reinterpret film footages and photographs obtained from the machine eyes. And along with the animation, I have tried 360-degree video. On the right side, you can see the slide. So this is Thai Moshan in New Territory of Hong Kong. So, and this is a dried valley that orchid species are living in the wild in Hong Kong. So the technique itself is not very new to anymore, but I employ this technique as a machine eye that overcomes the limitation of human eyesight and captures the whole surroundings, hoping that it also represents plant eyes. And even though I explore other possibilities of using camera and machine eyes, I have not been able to answer the questions about the role of machines in my practice. Keeping the unsolved questions aside, I turn to the research stage of researching seed germination. So here on the left side, this is how seed germination looks with a specific species called Liparis viridiflora. So on the very left, on the top side, that's the seeds that harvested from the capsule freshly. And then after 32 weeks, that's on the left down side, so it turns to be a little bit of green color. And on the right side, that's the photo of the seed germinated finally and will develop into a plant. I decided to visualize this process with algorithmic graphic. Unexpectedly, researching plant-inspired algorithms, I discovered that the application of machine learning to eco-art opens up new potential for multi-species interactions within contemporary ecological agencies. In this regard, I dig into the topic of ecology and machine. What's the way of that human, non-humans that include more than human living beings and machines? What could be the multi-species relationships of us? Random Forest project by Tön Karelsa gives me a clue to the way. Random Forest research project brings machine learning to bear upon the ecological dimension and inspires a fresh mode of learning from the environment. So prior to Random Forest research project, in 2018, artist-researcher Tön Karelsa conducted a fieldwork during his Ars Bioarctica residency in Finnish Arctic. He found out that the snow-covered rocks were identified as snowmobiles. And furthermore, the artificial intelligence did not recognize trees there. From this experience, Karelsa realized that the field of artificial intelligence generally did not acknowledge ecological elements and non-human species other than animals and plants. He pointed out that machine learning does not have a neutral perspective, since the training sets are created from what humans have pre-selected. So he raises questions. And is it reasonable to model artificial intelligence on human intelligence when so many other intelligence around us? And furthermore, should machines also learn directly from animals and plants? Thus, in Random Forest, Karolze combined an artificial neural network model and decision tree model to process and analyze environmental data with the hope that the scope of his algorithm can include both biotic and abiotic organisms. So here I edited his intro video to Random Forest research. So original video is about 20 minutes, but here I excerpted like about 2 minutes.
When you're talking about super intelligent AI that can make changes to itself, it seems that we only have one chance to get the initial conditions right. And even then, we will need to absorb the economic and political consequences of getting them right. But the moment we admit that information processing is the source of intelligence, that some appropriate computational system is what the basis of intelligence is.
Artificial superintelligence could be an existential threat to humanity. The problem is that once AI starts teaching itself how to fulfill its goals, you stop having control over its plans. And once an AI starts working out how to make itself better, it can get much smarter than you much quicker. So why should we be afraid of computers? Because ultimately, it's not people who control what purposes artificial intelligence is put towards. It's companies.
So he again asked this question: what does it mean if machines join animals and plants? They're on more equal level of awareness. With this research, he aims to let environmental machine learning learn from biodiverse environment, which does not situate at human-made level of species hierarchy. Environmental machine learning interacts with ecological data and trains algorithms with it. Environmental machine learning inquires the impact of AI that goes beyond human species. He conducted fieldworks to train environmental machine learning to interact with living organisms and the environment. Through the fieldwork and environmental machine learning sessions, the research project Random Forest investigates how and where environmental machine learning is impacted by human bias, as well as how machines can relate to multi-species relationships. So he conducted 4 sessions of fieldwork. The first session was conducted at Terschelling Island on the northern Netherlands. Environmental machine learning algorithms reflect and resemble the way humans see the environment, for instance, as a resources for industry. To investigate the influence of AI on the landscape, he regarded AI as an organism and see how AI impacts to the ecosystem. In the following session, he focused on finding a way with AI to give a nature to give nature a voice that is free from human interference. This session aims AI to learn directly from non-humans and surrounding environment. Therefore, artificial intelligence to be grounded in a self-identity that is independent of its creator, humans. Ideas developed into the third session, where he investigated AI as an algorithmic companion species in relation to non-humans. In his last session, he worked in the eastern Estonia and explored surrounding nature as an entity where AI can stay as a digital version of indigenous companion. He researched the way that AI can go beyond quantitative environmental data, but to be native in the environment. So in this regard, the researcher introduces artificial intelligence and machines, which could be considered as an extension of human species into multi-species relationships. I got a clue to how to situate the camera lens in the context of my research. Machine eyes that can be an extension of human eyes actually could mediate myself to the multi-species agency of orchids. More importantly, his research demonstrates the need to consider ecological correctness in the filmmaking process without species hierarchy and simultaneously overcomes the limitation of anthropocentrism. This principle is central to my own practice-based research, which tries to avoid human-centric gaze, but to learn from plants' perspective. Technological implications take an important role in working with art and multi-species. Utilizing machine learning in artistic practice enables artists not only to collect environmental data and shape them into different forms, but also to practice multi-species and computer relation and participate in the ecological community. It resonates with the concept of eco-cinema that I referred to. The Stefan Liga and Ina Bolinsky explained that human species can take a multi-species turn and move beyond human-centered perspective to encompass multi-species and non-human agencies in the surrounding environment. By collaborating with technologies in various fields, including art and ecology. The long history of the Anthropocene has unbalanced the holistic environment of multi-species relationships. So, researching the ecological engagement of machine learning supports the ethical participation of AI in multi-species relationships. So that was the end of my presentation. And to end my presentation, I may promote the exhibition that happens on 9th floor. The Conference Salon exhibition System Dreams is open until 10 p.m. during the symposium. So please feel free to come upstairs when you finish visiting 3rd floor exhibition. Thank you.
Thank you so much. Thank you so much, Taeyun, for a great presentation. Now we have 1 minute for our online attendees to type their questions via Zoom chat. The chat room will be closed after 1 minute, and we will start a question and answer session. And a little bit a remark about the question and answer session. I will voice the written questions on behalf of online attendees, while in-person participants can simply raise their hands if they have questions, and they will be given a microphone so that online participants can also listen. I think we can probably start with our first speaker because we have many questions online also. So maybe we can move on from one speaker to another. And there is one question to Georg Tremmel from Professor Lam Yun Wang. The question is, I would like to know more about how you incorporated the collected DNA into the iPS cells. Also, whose cells were the iPS cells derived from?
Yeah, so that's a very good question. So of course we needed to, so the iPS cells were from an anonymous donor of Japanese origin. So it was a cell line we bought. because this was the easiest way to do this. And the way they were incorporated was, of course, only a small bit of the DNA was incorporated, so we could not really. It was just a matter of price, so how much actual DNA can be synthesized and incorporated. But this was just done using a simple CRISPR array. Yeah, but we wanted to make sure that the DNA is not really expressed. It's just about representing the idea of this digital data encoded in biological DNA.
And these were human cells, yes?
Yes.
Mm-hmm. Okay,
yeah.
Thank you. Yeah, and anonymously you bought it from specific laboratory or just?
Yeah, so there are tissue culture banks, and this was the most straightforward way, because the challenge is also how to exhibit it, and if it would be a if it would be our own, if it would be a non-standard cell line, it would be much more difficult to exhibit them in an art space. And we could have exhibited them in a lab, but not in an art space. So this is always the problem when you work with genetic modification, that you really need to have, you need to prove that the material is safe to work with and to be exhibited in an art space. So I think this is, yeah.
Mm-hmm. Thank you so much. There's one more question. Would you mind to share more what do you mean by integrating synthetic DNA to machine learning models?
Yeah, so I quickly answered it in the chat already, but I think this was, I also shared a paper I was involved, which deals with nanobore base calling. So nanobore is a single cell sequencing technique, and we developed a unit-based method for how to sequence this, how to segment the pH data from the nanopores more correctly. And so this is a challenge, how to transcode the digital DNA into the machine learning model so that it makes meaningful images, or is it possible or not? So this is the unknown yet. But basically this is a lot of the core of media art, is this transmutation, transcoding of different data.
Mm-hmm. And there is one more question, which is a little bit wider, probably than the scope of the artwork you described. Mm-hmm. So it is how biohackers would avoid potential sufferings of the subjects.
That's a very, very good question. So, of course, when we established our bioart lab here in Tokyo, we made sure we followed the government guidelines. We also have an ethics commission, so any experiments needs to be approved by a board. And of course, the goal is to minimize suffering. But our lab is too small, so we only work with bacteria and cell lines. But even in universities, you need to prove that it's for the good of science before working or before inflicting suffering on animals. And we try not to do this. So that's why I prefer to work with cell lines. But the question is, if you work with organoids or like iPS-derived nerve cells, which you can grow in like three-dimensional structures, and they could potentially have consciousness. So there are other rules which are currently, I think, 2 weeks or depending on which legislation you are. So you only have a certain time to work with neurons, because there is the suspicion that they might become conscious and they might feel pain.
Mm-hmm. Yeah. Okay, thank you so much. Are there more questions from the audience for Georg Tremmel? Yeah.
So actually I have one question, because I heard that the Hatsune Miku, that's very huge in Japan. And I also a few years ago saw a news that a guy actually really did a wedding ceremony with the character. So I was wondering how the audience reaction about the heartbeat in the exhibition.
Yeah, no, I have to admit, although I'm living here, I'm not a super geek, so I'm not that much into the manga culture. But at the exhibition, it was really surprising, because as you saw, it was a big room, but they had to make like at concerts, like a way for people to wait, and then everybody could only spend 5 minutes there, the face pressed onto the screen. So and we had some people who actually went quite often there, So I maybe I should do more research into the pop culture phenomenon here. Yeah. But yeah, I was very happy at some point. I saw actually some people in the street in Tokyo with we also made a T-shirt, and I saw people with our T-shirts as well. So I was very happy that there was some impact on that.
I have a follow-up question on this because I think you also so the project. This project, this persona is collaborative. It is created collaboratively by people, but you also collected DNA genome kind of as a collaborative project online.
Yeah, I'm sorry.
Elaborate on it.
Yeah, I'm sorry. Maybe this did not come out so well, and maybe I didn't include the right slides. But the idea was to kind of apply the same procedure, or kind of open it up to the same procedure that people. So the image and the voice. People could experiment and play with it and collectively create this persona, and we opened up the same processes to actually directly editing digitally on the browser, editing the DNA. Of course, it would not make biological sense, but for example, what people did, they searched for genes which have a link to like green color, and they inserted this, so with the hope of creating a biological appropriation for this. But it was an exercise in speculation. So of course it's not. Yeah, the fact is we don't know yet how to create, how to rearrange the DNA as such. So this is why we also don't know how many cancers work yet. But it's a speculative idea, and that's why we took only certain parts of the DNA, which we are sure would not change the liveliness of the cells.
Thank you. Other questions? Yeah, I see a question from Maggie.
Hi. So, in terms of speculation, which we share with you as a methodology, what would you say underpins the idea of putting a heart, or the materiality, or the sort of reference to the materiality of a heart and all its symbolic and poetic and life biology gestures into a sort of virtual icon?
Yes, so maybe we went to the same socialization of speculative design, which is happened 10, 15 years ago in the UK, and is, of course, only done in Fiona Raby. And I was like, I had a chance to be like there when it was still proto-speculative. So we did not really have it wasn't coined yet, it was still critical design. But what I'm always suspicious about speculative design is that it creates speculative solution for speculative problems, and sometimes it's too much removed from the daily life. And what we try to do, we really tried. It was very important for us to not only simulate the heart, but actually to create an actual heart from real heart cells. From this, to actually collapse this idea of speculation to the present. And of course, what I'm doing now with the synthetic heart cells, it's another, maybe that's more speculative than the other one. That's why I'm curious of how to combine these two codes again.
But it's to get people to think about what's life, or like what's these kind of underlying concepts?
Yeah, of course. So I think many biological artworks deal with concepts of life. What does it mean to be alive? Is there life outside human bodies? What is the semi-living? And so we always had this idea of we don't give life, but to this character, but we could enable a journey between the digital and the biological worlds. So we have this idea of going back and forth and existing in different states. And this is maybe the poetic underpinning of this.
If I can just make a comment, I mean, I don't think this is the angle you were going on this work, but it struck me what was so interesting is its comment on iconology and on idols, and you're creating a sacred heart. You're actually bringing the heart to life of this idol, this figure. So it strikes me that the work has, although there's this biological, comment on biology and the virtual, but it has a much deeper cultural resonance than that. I mean, the resonance seems to be, the cultural resonance seems profound. I mean, you were creating a shrine for fans of this figure to come and worship or come and touch the sacred heart of this figure. It has extraordinary cultural resonance, I think.
Yeah, that's interesting how maybe the idolography, or how do you say idolatry of, I don't know what's the word, how this works in, of course, I grew up in Austria, so Central or European, we have a different way of looking at idols and artifacts. So maybe the Japanese culture and the Japanese religion or kind of Asian cultural sphere has different ways of dealing with that. But yeah, it's still amazes me that, so not only this kind of created idol, but we have examples. So William Gibson, he wrote a whole book in Idol, or many pop stars in Asian countries are elevated to the stage of idols. Yeah? So it's how much this is a substitute for religion is another question. Yeah? Or how much? So if they're worshipped, what do they give back? Yeah? Can they give something back?
There's one more question from the audience.
Oh, sorry. Can you hear me? Yeah, okay. I just wanted to comment quickly on what was just said, because even semiotically, I think it's interesting, right? Because you're creating an image of the heart with a heart, or with a synthetic heart, right? So, I mean, in terms of like the question of the indexicality of the image, right? That it is, so it's no longer even just a visualization through what the heart is producing, but it's actually, you're like, as you said, it's moving beyond the kind of the representational to this direct connection to the thing that it's representing, though the thing that it's representing is not the actual thing, because it's a synthetic heart. So I don't know, there's a really interesting way in which it's also playing semiotically with representation, questions of representation, digital culture, I think. Sorry, yeah, it's just a comment.
So actually people, so the Holy Grail is actually to make a three-dimensional heart, but then you have problems with the blood vessels and so on. But it just occurs to me that this is actually, because it's in a petri dish, so it's a two-dimensional layer of hearts, but it's still synced up. So maybe it's quite fitting for a two-dimensional character to have a two-dimensional heart.
Yeah. Are there more questions for Georg? Maybe we move on to our next speakers and maybe come back to Georg later. So there are questions in the chat for Stephanie and Maggie. So the first question is about the rewarding function for the reinforcement learning in the project. So the question is, the person writes, I'm a bit curious about rewarding function, which rewarding function was used in the reinforcement learning in the project?
Okay, so I'll answer that. The short answer is we haven't decided yet, because we haven't done it yet. The most speculative answer is that what's hard-coded as the equilibrium state is likely to be set at something that corresponds to a state of curiosity in the octopus. So that might, say from Maggie's research, and from her, in fact, if AI were cephalopod exhibition in San Francisco the other year, that might correspond to sort of orangey red color and increased movement, which is a sign of curiosity. And maybe I'll let Maggie talk a bit more about that, or the sort of chroma chatter and play. But to do that, we'd need to spend, say, the first month with the octopus and the AI, calibrating the sensors and making those kinds of decisions. And it depends, partly on the octopus. The octopus is a pretty individual yeah, they vary hugely between octopus and octopus, just as humans do. Maggie, did you want to add anything to that?
We'll be interviewing quite a few, though.
We will.
Well, I technically no. I can't add anything.
I was just thinking in terms of the, chroma chatter and play and curiosity.
Yeah, I mean, we're trying to avoid the traditional scientific ways of communicating with an animal, starting with the mesocosm being the most ethical kind of husbandry that doesn't have the octopus in a tank kind of being poked and given labyrinths where there's a crab at the end of it, etc. It's as natural as an environment as possible, apart from the lack of predators, which hopefully will be a plus. But the play, we want to create interactive things on screens where the octopus will probably learn to touch something and realize things are happening by what it's touching, and a lot of that will be based on color and shape and emulating things in the environment, and also objects, sort of puzzle objects, 3D printed objects with accelerometers in them. But none of it's really to get the octopus to do anything. So what Stephanie's saying here is so key. It's about its curiosity, and I don't know what to add. I hadn't an idea of what we're working we haven't discussed the two teams, how the ML will be prototyped. So you know way more.
Yeah, and I think, part of the thing is, like Maggie said, we want it to be, the importance is that it's a holistic system that the AI is learning from. So we don't want to put any of our sort of prejudgments about what might constitute curiosity for the octopus, although we can have an idea about that from our research, and what might constitute engagement, I suppose. So some of this will be worked out in response.
Mm-hmm. Thank you. I see one question from that Mariana wants to ask a question.
Yes. Yeah, I want to ask a question to Stephanie and Maggie. I wanted to ask you two things. First, how developed is the notion of octopi intelligence as a model for AI? Because it's like, as you say, is this embodied cognition that is distributed in a different way? So I like how developed is this notion of octopi as this model for artificial intelligence instead of, human intelligence as the model? This is like the first one, if you know how developed this is. And the second is, why do you use the screens for interaction? And if you consider like some other form of interaction with the organisms, maybe because we are a little bit, I would be a little bit tired of like the exposure to screens as my form of interaction with the world. So, and I also wonder, like in the way that octopi see the world, etc., why did you choose the screens as the means of interactivity with them, and what does it give you, instead of other media?
Can I go quickly first? So from the mesocosm image that Stephanie showed, it isn't just screens at all. It's all sorts of there'll be kinds of very delicate sound elements, like nothing that would seem like a bomb going off, obviously. Like there'll be light things, there'll be interactive 3D objects that maybe change color or shape, or not shape exactly, but could be used to build with, or depending on being touched. There'll be the screen thing was really it's one element that is easily modulated by an AI, as in the data can be. Well, I'll pass that over to Stephanie, but just to emphasize, we're thinking about tactile and light and dark and shadow and many different and textual things. We're thinking about a lot of stuff that will be in the metacosm, not all at once, but screens are I think was very intrigued by watching that film I mentioned, The Octopus in My House, though it's quite a weird thing to have done to an octopus, but it does seem to be registering screens and interested in the movements and patterns and stuff on it, because a lot of animals don't, like my cat doesn't register a screen as a space of interest unless there's a howling noise coming out of it. And I think that's that's just the animal I know a little of. I have no idea what, but with screens has got a lot of potential for different kinds of visual content, really. It's just one element, but I think screens are training us a lot, as a way at the moment of trying to think into octopus cognition a bit, because we really have done so much research on how their camouflage works, how the chemotactile levels of information are passed into the arms by the kind of 2,000 suckers on each arm. Anyway, I'll pass to Stephanie if you have things to add.
Yeah, thank you. Yeah, as Maggie said, it's the sort of means of feedback, if you like, for the octopus. Like it can have an effect on what is. It can modulate what is shown on the screens, and I suppose it's also a sign of our own human limitations. So our capacity to understand octopuses. And visuality is one of the key ways that where we converge, if you like. So we understand something about octopus vision, although they use vision in a different way to humans. There is some convergent evolution of the eye, but they also are thought to see through their skin. We don't know to what extent. They certainly pattern match with their environments. So yeah, it's the point of maybe most connection that we can make, as well as the point where we can see the feedback. And I suppose it's important to say that the screens and what they're screening, some of the imagery that we've shown today, some of those sorts of imagery, this sort of acts as an invitation or to respond.
And I suppose just really quickly, because you've definitely got us going there, is we spent I've been in South Africa during the lockdowns and learnt to free dive, because I just thought this is an opportunity here to try and enter octopus environment in quite a meditative, open way and get used to it. And I think been thinking a lot about, like, just adding to what Stephanie said about it's a place that we can kind of engage in, a screen, but also what we put on those screens that is really to do with pattern and rhythm and wave and pressure and event that is quite abstract in a way to a human sense of representation and looking for meaning. So the screen's definitely a place where we can learn as well, probably more than making 3D objects, because octopuses use so many different things they find in the ocean, some of which is human detritus, to build dens or hide in, or whatever they're doing, moving around on the ocean floor. So the screen's probably the point of most kind of transformative learning for us in a way, and where we hope to glimpse moments of modulation by the AI that I don't know. We don't know. The unknown. We're big on the unknown.
Yeah. Well, we do know that octopuses we certainly know anecdotally that octopuses like to watch TV if there's a TV in the room where they are when they're not in the wild. But the other part of your question about octopuses used for AI. So it sort of seems like we could learn a lot from octopuses in terms of distributed cognition for AI. So far as I know, no one's used octopuses in this way. Well, no one's used octopuses in this way before or worked with octopuses in this way before. But octopus arm movements have been studied, and there's a sort of standard algorithm for modeling Gaussian movement that's used in underwater robotics and and robotics for surgery, for example, that has been derived from octopus movement. So it seems like there's lots of other things that we could learn in a more sort of holistic way from them, and maybe less sort of human, sort of less sort of humanly instrumental way, if you like.
Have you seen Georg's comment in the chat, Stephanie?
There is one more question here from Peter Nelson. When you use 2D or 2D animation for visualization, what is the relationship between abstractions, procedural animation, colors, etc., and empirical data, mechanism of octopus skin, microscopic forms, etc.? Is it metaphorical, literal, or a combination?
Shall I quickly start?
Yeah,
no, you go.
Well, I mean, Maggie, the Open Drift team has been doing this. So this is, like, in a sense, the first part of the invitation based on our own visual research and research into octopus cognition. And certainly the team are using algorithms like Turing's morphogenesis algorithm on animal, about how animal camouflage changes. So yes, that's a direct link to modeling actual camouflage skin changes. But there's also, I mean, maybe Maggie will talk about this, there is also more of a sort of imaginative, metaphorical level to it.
And also, another thing in our interdisciplinarity that we're thinking moving forwards into the future, science needs to be in communication with people who have a different way of knowledge gathering in the world, and which would include, like there's an amazing paper on the Serpentine's Creative AI Lab database called Indigenous Protocols for Machine Learning, which is worth the read. And then I've been working with a renowned interspecies communicator who uses visualization techniques and has kind of done work in quantum physics to try and help how she visualizes, and it's treated as very speculative, but the information we got to do with not how camouflage works, that the relationship between iridophores, leucophores, chromatophores in the skin are obviously scientific fact, what did you call empirical, but then how that operates, how the octopus is becoming its environment, is in a state of mimesis with the surface it's on, or the textures, or whatever. That came through animal communication mentoring, and I'd say it's a combination of those two. That's empirical for me. Like, those two are equally important ways of knowledge gathering. And then there's the imagination, as Stephanie mentioned. But the art team have been doing the individuals involved, like didn't have time to show you lots of Megan's work where she's literally worked on what the 3 kinds of camouflage cell do in the skin, and then interpreted that through the coding images that I showed you, the procedural Voronoi combination.
Thank you so much. I think we now should probably move on to our next speakers and the questions for the next speakers, since we have 2 more speakers. I would like to ask, are there questions for Mariana, for Maro Pebo in the audience? Everything is clear. Yeah, probably because we all saw your work, so yeah, therefore there are less questions. I would like to ask one question about your work. I really like the narrative and the visuals. And you also mentioned it in your presentation that you go beyond the common scientific visualizations of mitochondria and more kind of biological visualizations. Could you elaborate more on this choice?
Sure. I'm very interested in scientific representation and what it means, how it conveys meaning outside of what it's in the discourse. For example, in a research paper, there's always images and they also convey meaning that we take for granted. So I wanted to play with this layer of meaning and how to change a little bit the way that we relate to organisms as this scientific object, and how to think about it as a more like mythological origin of ourselves, you know, like constructed as a narrative. And also in terms of the aesthetic, we were thinking about something that was very, very timely and ephemeral. And that when people would watch this, they would see this was made in 2020. You know, there was no confusion. And that comes from my approach as an art historian, in which I enjoy being able to tell when an artwork comes from just by like the aesthetic choices that they have made. So like that's why we went for this very millennial pop kind of visual culture, you know, so people would identify with it, and also so it could also function as an object in itself, like this form of producing an experience for an audience that would tell me that the mitochondria is boring, but that could enjoy this more kind of fluid eye candy narrative, even if you don't know what's going on really. So that's a little bit the aesthetic choices that we made for to the storytelling. But yeah, thank you for your question, Olga.
Yeah, and I have one more question about your interest in symbiogenesis. In general, maybe you could talk a little bit more on the context. And the follow-up question connected to it is that you work with extraction of mitochondria, and I'm curious why, talking about symbiogenesis, you make a choice of extraction, and as you mentioned, like violent extraction and violent emancipation. So why do you choose this kind of direction in talking about symbiogenesis, which is sort of counterintuitive, and probably that's why very interesting, yeah.
Yeah, which is like exactly like the opposite, right? Yeah. So I'm interested in symbiosis in general, how we establish relationship with the others, a little bit like this panel, like these interspecies relationships. As I said in the beginning, I'm interested more in these trans-species relationships. I believe that this structure dividing by species these taxonomical divisions are very artificial, let's say, and do not sustain very well, especially when it comes to microorganisms with their, horizontal gene transfer, etc. And of course, I'm mostly interested in symbiogenesis as a way to identify ourselves in some form as trans species from the outset. Right? And so this is about like putting things together. And in fact, yes, this artwork is about separating them. So that's a very difficult but good question that you're asking there. So I wanted to, actually my like speculative dimension of the work was, what if we could release the bacteria to be free again? That's why it's called emancipation. What would happen if we could release the mitochondria for them to function as free bacteria again. And this would be like the ultimate proof for everyone to realize how deep this relationship is. Then would be like, whoa, is there bacteria living in me? You know? So that's why I decided, this was like the starting point, you know, to break the symbiosis, to prove it. You know, that's like, and that brings us to the notion of sacrifice, which is also very important for me. You know, I'm going to sacrifice the cell, and I'm going to sacrifice myself and my blood, and to prove the symbiosis, you know, to bring it forward. This is something that took place a long time ago, and it's part of my existence every single moment of my life for me and every plant and every animal in the world. And in order to prove it or to bring it forward, I'm going to sacrifice it for this one time. So this, like the notion of sacrifice and the sacred and the religious that comes forward also in the notion of the reliquary, etc. But it's actually a paradox that the artwork proposes, you know, like breaking the symbiosis, the unity, in order to prove it and to bring it forth and to highlight it. Thanks.
Thank you. Are there more questions for Mariana, for Maro? Yeah. Maybe I ask one more question and then we move on to the next speaker. So I'm curious also, because I think you are also interested, as many as speakers today, most of the speakers today, in the post-anthropocentric kind of narratives and searching for ways to deal with different species in a post-anthropocentric way. And again, there is another very interesting counterintuitive thing in your work about this sacred microbial origin, and also that the way you present the mitochondria. So I'm curious that this is a very kind of anthropocentric way of presenting the work. And I find it very interesting because probably this is how we as humans can understand it better. But could you also elaborate on this kind of controversy?
Mm-hmm. So the artwork is always for a person, you know, like I never pretend that the artwork is for the organisms or whatever. These artworks are clearly for humans, you know, for humans to enjoy and for humans to understand, and the discourse and the narrative I'm building is for other humans to understand their own identity. So in this, I don't see so much the conflict. What I think is interesting for me is when talking about the post-human was to be as radical as possible. So for example, I really find it fascinating the work with octopus because they're so incredibly smart. You know, we can right away like perceive their intelligence. So in terms of post-human thinking, this is humbling right away, you know, as we thought we were like holding the monopoly of cognition, and octopus is just like, you know, makes us change our mind right away. But I wanted to take it to the most radical place, and for me the most radical place are microorganisms. As Georg was saying before, you know, we can do whatever we want with them in the lab. There's no ethical concerns. And what I would ask, like, why, you know, where do we draw the line on like which organisms it's okay to tamper with and play with and torture and which ones not, you know? So I want to take it to the most like radical, minimal expression of life when we're facing the otherness, you know, not the other human ethnically or gender-wise, but like the other species in the most minimal expression of what we consider to be a living organism in this kind of very simple bacteria, let's say, you know, or even when we work with viruses that we're not even sure that they're alive. So this is like this radical otherness that I wanted to play with, and when thinking about the post-human, and that's why I chose these organisms.
Thank you so much.
I'll just make a comment. So of course, thank you, Maro, for your great talk and your comments. So of course, we all living things, we are related, right? Because we share the same genetic code. So of course, this is one of these mind-blowing realizations. And of course, the other thing is that I'm sure everybody here knows this, but the biggest catastrophe in the Earth history happened like about 3 billion years ago, right? And this was the great oxidation event, which was triggered by cyanobacteria, and it basically killed 99.999% of life on Earth by releasing oxygen in the atmosphere. and basically killing everything. But this is also the reason why we're here. Yeah, and so this kind of idea of minimal life and what is original life, and that's a very, very interesting question for all of us. Thank you.
Yeah, I love the idea of the great oxygen catastrophe. What is catastrophic for some is beneficial for others. I think I'm very happy you brought it up. And also, of course, we all evolved from microorganisms, but there's like these secondary events, like the chloroplasts, for example, that when they came, or the mitochondria came. This is beyond the evolution from, where we're coming from, but these extra moments of relation, of symbiosis that are interesting to me. But I think we should go ahead and talk to Jade. Yeah,
so make a microbiome of robots. Maybe that's something we can do in the future.
Exactly. Or organelles that are all like external, synthetic, but we're already working on that. So, yeah.
Yeah, thank you so much, Maro. And now we're moving on to the questions for Tiyun. And there are 2 questions in the chat. One question is, what is your preferred technique, animation or 360 video?
Oh.
From Eugenia Kim.
I do like them both, but initially I thought, when I really started this research project, I thought 360-degree video would be more effective for my research, but now I realize that I am realizing the limitation of 360-degree and visualizing the plant and multi-species interactions happening there. So I would say the combination of animation and 360 video is my preference. Because I think those 2 complement each other.
Mm-hmm. Thank you. I hope Eugenia is satisfied with the question, with the answer. And now we have the next question from Louis Mancun. The question is, could you share a bit more about what type of data, quantitative sensory, does the artist collect from the environment, and how are the data represented?
So the artist, he really wants to create the algorithm that let artificial intelligence to learn directly from the environment. So that's why I mentioned there's 2 different algorithm models that he combined to create his own algorithm. And so he collected both quantitative and sensory data from the environment, and he actually represents those things in a very different, many different types of way. And to introduce all these things, it may take a longer time. So, but generously, the artist shared everything, literally from A to Z, on his website. So, on his publication only about the fieldwork and the sharing sessions. So, I can share the link with Mancun. Yeah.
Okay, thank you. Are there maybe questions in the audience for Tiyun? Yeah, I'm curious about how you're interested. Are you interested in working with artificial intelligence in your own project, and how exactly you might, if you're interested, how you might use it?
So actually, this algorithm and AI topic came to my research very recently, and that's because I wanted to find a way to situate the role of camera and that machine eyes into my research. And now I'm really interested in developing this algorithmic generative video, moving image, with the seed germination that I explained before. And I am very new to this field. I'm still researching and learning more. So, but I am definitely interested in machine learning in the ecological sense. So I may be able to employ some of them to my research.
Thank you so much. Are there maybe some more questions for Tiyun online from our presenters? If no, then we can move on to final remarks. If you have some final remarks, I would like to share information about your future work. We would be very interested to learn more. For instance, Mariana, if you are going to develop your project somehow and how.
Hey, well, all my projects are with microorganisms, but thinking about symbiosis and endosymbiosis, I'm very interested in trying to find ways to work exactly with chloroplasts, because the relationship with cyanobacteria allows for photosynthesis in all plants, which also basically transform the face of our planet. But these cells are way harder to break into. But I would love to explore the notion of the chloroplast and the cyanobacteria and the relationship that we establish with them. So this is something that I would love to work on, and also the idea of symbiosis, but in between different species of organisms. So I work with Winogradsky columns in my research, but now in my practice, as these communities of interspecies organisms interchanging genes and metabolites and everything. So these are like the projects I'm interested in for the future.
And I assume, I don't know, do you have more remarks, Maggie, Stephanie, about your future work? Maybe not necessarily about the work with octopus and AI. No?
It's a long project.
Mm-hmm.
It's going to then be in art exhibitions and working on with what kind of interactive technologies we use. I mean, it's just sort of at the moment feels like a good many years are evolving through this. I don't know about Stephanie.
Yeah, same.
Mm-hmm. Yeah, it seems it will take a lot of time to actually make it work in all the steps you want to do, as you mentioned today. And
may I? I'm sorry.
Yeah.
Sorry, may I make a final? So the research progress of my work will be exhibited in Hong Kong. So people who are present in Hong Kong, please visit JCCAC Level 01 Gallery in July to check out. Thank you.
Sure. Yeah. Maybe some final remarks from Georg.
So not really remarks, but Maro, so our lab at the Metaforest at Waseda is actually a cyanobacterial lab, so run by Hideo Iwasaki, and we actually also have artists in residences. So we're very happy to. So this is, of course, to everybody. So please come to Japan if the corona allows it, and please let's collaborate with Metaforest and also with BioClub. Would be very happy to get more collaboration and conversation going.
Thank you. Thank you a lot, all the presenters, for your great presentation and your responses to the questions. I would like to thank also the audience, online audience and offline audience. And we are finishing the session now, and there will be dinner for people here. Yes, I hope to see you all somewhere soon, hopefully in person. Yeah. Thank you again. Bye-bye.
Bye.
Thank you very much. Bye-bye.